In this video, we are going to discuss about prokaryotic transcription and what are the enzymes present in this and what are the steps and the significance of the transcription. Let us move on to transcription. What is transcription? It is the process by which the information in a strand of DNA is copied into a new molecule of messenger RNA, that is mRNA. So the information from the DNA molecule is copied into an mRNA molecule. This is known as transcription. So the transcription is the process by which the information in a strand of DNA is copied into a new molecule of mRNA. In prokaryotic organism, transcription occurs in three phases. They are initiation, elongation and termination. Transcription occurs in three stages: initiation, elongation and termination. Now, let us say what are the enzymes present in the transcription. The most important enzyme in the transcription process is RNA polymerase. RNA is synthesized by a single RNA polymerase enzyme which contains multiple polypeptide subunits. This RNA polymerase contains multiple polypeptide subunits. In E. coli, the RNA polymerase has subunits 2 alpha, 1 beta, 1 beta dash and 1 omega and 1 sigma subunit. Contain 2 alpha, 1 beta, 1 beta dash, 1 omega and 1 sigma. This complete enzyme is called the holo enzyme. When the sigma subunit dissociate from this enzyme, they are known as core enzyme. The sigma subunit dissociate from the enzyme, then they are known as core enzymes. So the first step is initiation. During the initiation, the RNA polymerase recognizes a specific site on the DNA that is upstream from the gene that will be transcribed and is called promoter site. Then unwinds the DNA locally. So the first step is the recognition of the promoter site by the enzyme RNA polymerase. So the RNA polymerase first recognizes the promoter site and it starts the DNA unwind locally. Now let us discuss about promoters and initiation. The holo enzyme binds to promoter region that is about 40 to 60 base pair in size and then initiate the transcription a short distance downstream. That is, if it is the DNA strand, here the promoter, then the RNA polymerase binds to the promoter and then starts the transcription. And within the promoter, there are two 6 base pair sequences. They are particularly important for the promoter function and they are highly conserved between species. So, using the convention of calling the first nucleotide of a transcribed sequence as +1, these two promoter elements lie at a position -10 and -35, that is about 10 and 35 base pair upstream of where the transcription will begin. That is, if it is the DNA to be transcribed, here this is the promoter site. In this promoter site, there will be two 6 base pair sequences. If the start site, we can consider the start site as +1. So if this is the +1 site, here the transcription start and this two base pair sequences, that is two 6 base pair sequences, they are known as -10 sequence and -35 sequence. This -10 sequence will be here, that is -10 base pairs upstream, and minus 35 will be here, that is 35 base pair upstream from the transcription start site. Once more, if we can this is the promoter, and this is the DNA to be transcribed, and this is the promoter site. So in this promoter there are two 6 base pair sequences. They are known as minus 10 sequence and minus 35 sequence. So they located on 10 base pair from the start site, and that is known as minus 10 region, and 35 base pair from the start site upstream of the DNA, that is known as minus 35 region or minus 35 sequence. And the start site we can consider as plus 1. So here will be minus 10 base pair upstream will be the 6 base pair sequence, one that is known as minus 10, and minus 35 upstream there will be another 6 base pair sequence, that is known as minus 35 sequence. Usually these are important for the promoter function. The minus 10 sequence has consensus, and the element was discovered by Pribnow. So it is known as Pribnow box. So the minus 10 sequence is known as Pribnow box. It is an important recognition site and that interact with the sigma factor of the RNA polymerase. So when the RNA polymerase bind to the promoter site, the sigma factor bind to the minus 10 sequence, and minus 35 sequence, it has a consensus TTGA-CA sequence, and it is important in DNA unwinding during transcriptional initiation. This involved in the unwinding during the transcriptional initiation. So the minus 10 sequence involved in the binding of the sigma factor of the RNA polymerase, and minus 35 sequence is a consensus TTGA-CA, and it is mainly function in the unwinding of the DNA during transcription initiation. As we discussed in the DNA replication, the transcription does not require a primer. So the RNA polymerase does not need a primer to begin the transcription, and which having bound to the promoter site, and the RNA polymerase begin the transcription directly. So there is no need of the primer in the transcription, but replication require the primer. Next we can discuss elongation phase. After the transcription initiation, the sigma factor is released from the transcriptional complex to leave the core enzyme, which continues elongation of the RNA transcript. So after the initiation, the sigma factor will release, and the core enzyme continues the elongation of the transcript. And the core enzyme contains the catalytic site for polymerization, probably within the beta subunit. The core enzyme contains the catalytic site for polymerization, that is probably in the beta subunit. The first nucleotide in the RNA transcript, usually guanine nucleotide or adenine nucleotide. The first nucleotide in the RNA transcript is usually guanine nucleotide or adenine nucleotide. The RNA polymerase then synthesizes the RNA in the 5' to 3' direction using the 4-ribonucleoside 5 triphosphates ATP, CTP, GTP and UTP as the precursors. So the RNA polymerase uses ATP, CTP, GTP and UTP as the precursors to elongate the RNA. The 3'OH end of the growing RNA chain attacks the alpha phosphate group of the incoming ribonucleoside 5 triphosphates to form a 3'5' phosphodiester bond. So, a 3'5' phosphodiester bond is formed between the 3' end of the growing RNA and the incoming ribonucleoside 5-phosphate. And the complex of RNA polymerase, DNA template and new RNA transcript is called ternary complex. Both these three are known as ternary complexes. And the region of unwound DNA that is undergoing transcription is called transcription bubble. So, where is the transcription start and where is the unwinding of the DNA takes place, that region is known as transcription bubble. The RNA transcript form a transient RNA DNA hybrid helix with its template strand and peels away from the DNA as transcription proceeds. That is, consider this is the DNA to be transcribed. Here the RNA polymerase binds and the mRNA is synthesized from here like this. As the RNA polymerase moves, it will synthesize the mRNA, and this mRNA and DNA form RNA DNA hybrid helix here. When the transcription proceeds, it will peels away from the DNA. And the DNA is unwound ahead of the transcription bubble, and after the transcription complex has passed, the DNA rewinds. If the transcription continues, the transcription bubble is formed by the RNA polymerase, and after the completion of the transcription of that region, the DNA rewinds. That is, if this is the double-stranded DNA, here the transcription proceeds here. So, here a transcription bubble is formed. Transcription bubble is formed here. And when the RNA polymerase move from here to here, this double strand will rewind. The unwound strands will rewind. At the same time, the bubble will move to here. So, when the RNA polymerase moves, there will become the transcription bubbles. So, that region become transcription bubble, and after the transcription complex has passed, then the DNA rewinds. Thus, during the elongation, the RNA polymerase uses the antisense strand of the DNA as template and synthesize a complementary RNA molecule that we will discuss later. So, the RNA produced are the same sequence as the non-template strand called the sense strand, except the RNA contains uracil instead of thymine. Now, let us discuss the process in details. Consider this is the DNA to be transcribed, and this DNA contains two strands. This is one is coding strand and the other one is non-coding strand. Non-coding strand. Coding strand is always known as non-template strand. Non-template strand. non-template strand. Coding strand is also known as non-template strand. It has also another name, sense strand. So, the DNA contains a double helix. In this, one strand is known as coding strand or non-template strand or sense strand, and the other one is known as non-coding strand or template strand. Template strand or antisense strand. Antisense strand. So, this is the strands, that is coding strand and non-coding strand. Non-coding strand also known as antisense strand or template strand, and coding strand also known as non-template strand or sense strand. Here we can use the non-coding strand as the template. Non-coding strand is used as the template by the enzyme RNA polymerase. Let us discuss the reaction. So, this is the double-stranded DNA. Here, one is coding strand and the other one is non-coding strand. So, this non-coding strand acts as the template and the coding node act as the template. So, the newly synthesized mRNA will be complementary to the non-coding template strand. The newly synthesized mRNA sequences are complementary to the non-coding template strand. Now, let us discuss the reaction. In this DNA, here is the transcription start site. So, in this, consider this is the template strand. So, in this template strand, in the upstream region, there will be a promoter site. There will be promoter in the template strand, that is upstream the start site. So, the RNA polymerase, which contain subunits 2 alpha, beta, beta dash, omega and sigma. So, the RNA polymerase scan the promoter site, and this promoter contain two sequences, 6 base pair sequences. They are -10 sequences, also known as box, and -35 sequence. These two sequences lie in between the promoter region. So, the start site we can use as +1. So, from here in 10 base pair away we can see the -10 region, and 35 base pair away upstream we can see -35 region. Once more, this is our DNA to be transcribed. Here consider that this is the start site, that is +1 site. From here the transcription start. So, to start the transcription, the RNA polymerase want to scan the promoter site. So, the RNA polymerase recognizes the promoter site, and that promoter site will be upstream of the +1 site, and in that promoter site there will be 6 base pair sequences. 6 base pair sequences. First one is -10 sequence or box, and the second one is -35 sequence. So, the -10 sequence is 10 base pair upstream from the start site. 10 base pair upstream from the start site. Similarly, the minus 35 sequence, that is 35 base pair upstream from the start site. So, this present in the promoter region of the template strand. When the RNA polymerase recognizes the promote site, it binds to the promoter region and start the transcription from the start site. When the RNA polymerase binds, the sigma subunit of the RNA polymerase, we already discussed that the RNA polymerase contain different subunits, 2 alpha, 1 beta, 1 beta dash, 1 sigma and 1 omega. So, these sigma subunits bind to the transcription, binds to the promoter site and starts the transcription. So, when the transcription initiates, the sigma subunits will dissociate from the RNA polymerase. Then the core enzyme will continue to add the nucleotides and to proceed the transcription. Now, the RNA polymerase does not require the primer, and when it binds to the promoter site, it starts to add the nucleotides and begins the transcription directly. Now, this is the RNA polymerase. When the RNA polymerase moves in this direction, that is 5' to 3' direction, the downstream DNA will unwind. That is here when the RNA polymerase moves, it will start to unwind the DNA. So, when the RNA polymerase moves through this, unwinds the DNA and add the nucleotides, and they are like this, primary transcript are like this. So, when the RNA polymerase moves, the primary transcripts elongates, and when the RNA polymerase moves, the position where the transcription is completed, where the DNA double helix will rewind. That is, when it moves forward, there will be unwinding, and after the completion, there will be rewinding. So, when the transcription completes, that position the DNA will rewind. And the elongation proceeds by the addition of the ATP, CTP, GTP and UTP. And this complex of RNA polymerase, transcript and DNA template are known as ternary complex. So, here the RNA transcript form a transient RNA-DNA hybrid helix with its template, but it peels away from the DNA as transcription proceeds, and the DNA unwound ahead of the transcription and rewound after the transcription. And this transcription continue until a termination sequence is reached. Usually the common termination sequences are GC rich regions. That is a palindrome. that is followed by an AT-rich sequence. GC-rich region and followed by an AT-rich region that forms palindromes. So the RNA made from the DNA palindromes is self-complementary. The RNA made from the DNA palindrome will be self-complementary and they will form hairpin-like structures, that is rich in GC base pairs, followed by 4 or more uracil residues. And not all termination sites does not require the hairpin structure. Instead of that, there will be some protein, they are known as raw protein, which helps to recognize the termination site and stop the transcription. Thus, the RNA polymerase encounters a termination signal and stop the transcription, releasing the RNA transcript and dissociating from the DNA. Once more, we can discuss the termination phase. The transcription continues until a termination sequence reached, and the most common termination sequences are GC-rich regions, that is palindromes, followed by an AT-rich sequence, and the RNA made from the DNA will be palindrome, so it's self-complementary. So base pairs internally to form hairpin structures in GC base pairs, followed by 4 or more uracil residues. And not all termination sites have this hairpin structure. Those that lack the structure requires an additional protein called raw protein, which helps to recognize the termination site and stop the transcription. Thus, the RNA polymerase encounters a termination signal and ceases the transcription, releasing the RNA transcript and dissociating from the DNA. This is the figure. This is the DNA helix. You can see here, this is the sigma factor inside the RNA polymerase, which will binds to the promoter region. This is the initiation step. After binding, the RNA polymerase will move and a new RNA transcript is formed here. And before that, the sigma factor is released before the elongation step, and then the core enzyme moves, RNA polymerase moves and add the nucleotides and forming a primary transcript. And this transcription continues till it reaches a termination sequence. Termination sequence may be palindrome sequences of GC-rich and AT-rich regions, and they will form hairpin-like structures, and the termination of the transcription will occur. And if there is lack of this termination sequences, some proteins known as raw factors act as the termination signal, that is, they recognizes the termination signal and transcription stops. This is the overall structure of the transcription, that is, this is the first step, initiation. In the time of initiation, at the primer region, at the promoter region, the RNA polymerase will bind and starts the unbinding of the DNA, and during the elongation time, the sigma factor from the RNA polymerase will dissociate, and then the core enzyme will continue to add the nucleotides and to form a primary transcript like this, and the nucleotides are added by the RNA polymerase from the 3' to 5' end, and the primary transcript will grow as the RNA polymerase moves. When the RNA polymerase move ahead of the DNA, double strand will unwind, and after the transcription it will rewind. Then this elongation occurs till a termination sequence reach, and when the RNA polymerase recognizes the termination sequence. That will be palindrome sequences of GC-rich and AT-rich regions. So they will be self-complementary and form hairpin structures and stop the transcription. At the same time, if there is no palindrome sequences are there, so the rho factor will help to stop the transcription. So the transcription is the process by which the information in strands of DNA is copied into a new molecule of messenger RNA, and there are 3 steps in the transcription: initiation, elongation and termination. And enzymes are RNA polymerase, mainly the enzyme RNA polymerase, and which has 5 subunits: 2 alpha, 1 beta, 1 beta dash and omega and sigma. And the sigma subunits dissociate from the subunit when the elongation starts. Then there will be promoters, and in the promoter region the RNA polymerase bind, and in the promoter region there will be 2 sequences of 6 base pairs. One is -10 sequence and the other one is -35 sequence. That is -10 will be 10 base pair upstream from the start site, and -35 will be 35 base pair upstream from the start site. Then the 35 sequence has consensus TTGA-CA sequence, and next one is the elongation phase. During the elongation phase, the RNA polymerase will bind and add the nucleotides, continues the elongation to form the RNA transcript, and the 3'OH end of the growing RNA chain attacked by the alpha phosphate group of the incoming ribonucleoside 5'triphosphates to form 3'5' phosphodiester bond. Then this complex, that is DNA template, new RNA transcript and RNA polymerase, these are all known as ternary complex. Then as the RNA polymerase moves, the primary transcript will form, that will form a DNA RNA or RNA DNA hybrid helix. When the transcription proceeds, it will peel away from the DNA, and the DNA is unwound ahead of the transcription bubble, and after the transcription complex has passed, the DNA rewind. And the elongation continues until it reaches a termination sequences, and the termination sequences will be contain GC-rich region or AT-rich region, that will be palindrome. So they will be self-complementary. So they will form hairpin-like structures. This will stop the transcription. If the self-complementary or palindrome sequences are absent, in that case a protein known as rho factor, which recognizes the termination site and stop transcription. This is all about transcription. Thank you.